Hello students, now we will learn about lysosomes, one of the cell organelles in cells. So lysosomes are discovered by Christian D. Dewey. Lysosomes are also called suicide sacs or suicidal bags. These lysosomes have more number and more types of autolysing enzymes or digesting enzymes. Next, these lysosomes are present in all types of eukaryotic cells, but there are some exceptions. Lysosomes are present in all eukaryotic cells except yeast, neurospora, euglena, meristematic cells, and a matured mammalian RBC. Next, number of these lysosomes, it vary according to the function. Number of lysosomes are more in WBC because they involve in phagocytic function, and osteoclasts. Osteoclasts are the cells that remove the aged osteoblast, means bone cells. Next, shape of lysosome is circular or spherical. Or spherical in shape. So we can draw, if this is a circular structure, inside will be the matrix and outside will be the memory. Next, there are different types of lysosomes based on the type of matrix. So lysosomes are said as polymorphic organelles. There are 4 types of lysosomes based on content, type of content of matrix. Since there are 4 types, we can say lysosomes are polymorphic organelles. So we will see what are those 4 types. First one, primary lysosomes. Since 4 different types are varying based on nature of content of the matrix, first one, primary lysosomes, they have inactive form of digesting enzymes. Inactive, we will write hydrolytic enzymes. Next, second one, secondary lysosomes. Secondary lysosomes. Now, these have the activated form of digesting enzymes or hydrolytic enzymes. These have active form of digesting enzymes. Next, third one, tertiary lysosomes. Tertiary lysosomes. These are also termed as residual bodies and have only undigested material. Means here, by the time of formation of tertiary lysosomes, the function of these digesting enzymes has been finished and the lysosome now is filled with the waste material. So, right, also called as residual bodies. By the time of reaching this stage, digestion by enzymes is finished and lysosomes have waste material inside them. Material to be exited. Now, by the time of tertiary lysosomes must do the exocytosis to remove the waste material from their internal matrix. Fourth one, autophagosomes or autolysosomes. Autophagosomes or autolysosomes. So, by the time they reaches autophagosome, now they are termed as suicidal bags. They digest self organ. They themselves gets damaged. So called as suicidal bags. Here, primary lysosome. Primary lysosome, it plays initial role in endocytosis. Initial role during endocytosis. Endocytosis means initially after releasing from the Golgi body, from the transit, these lysosomes will intake the food material that is entering through the plasma membrane and forms into a vacuole. For suppose if this is a plasma membrane and food material are being entering, then this area forms into a vacuole-like structure by the process of elasticity. So the food material enters inside. Now here readily primary lysosomes are present. So they will come and bind with this food vacuole that is formed. Now this will be the food vacuole. This will be the lysosome, primary lysosome. Together forms into one single layer and these food particles will enter into primary lysosome. So that is the role taken by primary lysosomes, that we are giving a term like endocytosis. Means intaking of food material and mixing with that of enzymes. We will see what are enzymes that are present in lysosomes. Enzymes within a lysosomes are proteases. Next, glycosidases, nucleases, next esterases, phosphatases, sulfatases. So under nucleases, both RNases and DNases also are studied. Next, coming to some of the functions of lysosomes apart from endocytosis and exocytosis, lysosomes are also involved in heterophagy. Heterophagy means digestion of food that is taken from outside and some other functions of lysosomes are heterophagy. Heterophagy means digestion of food that is taken from outside. Means digestion of food taken from outside. Then, what is called as digestion of reserve food material, that is called autophagy. Digestion of reserve food, that is called autophagy. So lysosomes are also doing heterophagy and autophagy. Next, they also destroy the carcinogens and supports in defense mechanism of the cell. Supports breaking off or destroying of carcinogens and helps in cell defense mechanism. Now, these three cells, primary, secondary and tertiary, they are involved in heterophagy. So these three are called as heterophagosomes. Heterophagosomes. Because they are involved in digestion of the food that is taken by the process of endocytosis and removal of the waste by the process of exocytosis.